So first of all, when you study fruits and vegetables, we know that there's a high variability. It means like there's difference in genetics, planting condition. For example, this farmer planted this in Spain while the other one planted it in Germany. Of course, those, even if they're the same strawberries or cucumber, they have huge variance because of those different conditions, and most of all because of post-harvest handling. So post-harvest handling, as we know, is like after the harvest. For example, the market, before it reached the consumer. So these are the post-harvest handling. And most of the producers and even the consumers, they waste a lot of fruits and vegetables because of post-harvest handling and consumer handling. So in my research, because of this high variability, in my research as part of the Serenade project, I am doing a lot of analysis, or we call it replicates or replications, so that we will lessen the variance. So in this case, I studied the physical-chemical properties, for example, the color, the texture of your strawberry, how the size, the shape, and also the chemical properties, the level of sugar, how sweet it is, the level of acidity, how sour it is, and also microbial properties. And most of all, as part of the Serenade project, we studied the volatile organic compounds, or VOC. So in simple terms, VOC, these are the gas that these fruits or vegetables emit, and then we use this data, these parameters, in order to predict and to create a model to predict the shelf life of these fruits and vegetables.
So, as a microbiologist point of view, I would say they are quite different, because when you think of fresh produce, mostly this comes under fruits and vegetables like that. And when you think of that, the most of the microbial factors are depending on the environment actually. So most of the microbes you are seeing on fruits and vegetables are from the environment, and for some vegetables it could be from soil too. But if you think in the case of cooked food, it's completely different, because you're cooking it, so the heat process will significantly lower the microbial load actually. They might be killed in the process of heat processing. But whatever occurs after that is because of post-cooking contamination. Let's put it like that, because it depends on the person or the individual who is cooking, and based on their hygiene practices, and also it depends on the how you are cooking, what kind of process you are doing in the kitchen. Like it could relate with your inadequate cooking process, and also because of the storage, how you are storing your food in the fridge like that. And when you would like me to talk about the key indicators in my research, so when you think of this cooked meals, and it depends, as I told before, it depends on the individual. So every kitchen is not the same, and so the cooking process. So they are highly variable, and so the microbial load. So I am keeping in my research in a broader aspect. So for the microbiological quantification, I am analyzing or focusing on broader range of spoilage microorganisms, and also I check the pH of the food, because some microbial growth can affect your pH of the food, and so it can cause some influences. And also visually we see how your food is transforming in the storage period. And last but not the least, the volatile organic compounds, which is produced by the microbes. So we will also evaluate them and try to correlate them. When microbes started to grow in your food, they tend to produce these volatile compounds. And when they all started to produce through a threshold, you can start to feel some smells in your food. That's the thing we are labeling as spoilage. So I think it's clear. So now the benefits. So when you think of these conventional methods, like traditional methods for analyzing microbial spoilage, they are time consuming, I would say. And you need some skilled persons like microbiologists to do an analysis. So it's going to take some time to know the results, what kind of microbial contaminations are present in your food, and what is going to cost to you. But when you are thinking of this microbial volatile compounds, and and the application towards the sensor technology, it could be really fast because you will just train your sensors for this detection of particular particular volatile compounds or maybe a broader groups, and you can just going to keep it in your food packets or containers, so instantly you will get the results. So I would say that the benefits, they are faster and can be accurate.
These VOCs, the main VOCs that I use as a biomarker in order to know when the fruit or vegetable will spoil is hexanal. So this hexanal is a specific chemical that this fruit or vegetable release that signals when it becomes spoiled or you cannot eat it anymore. So which means this hexanal will increase through time. For example, you store your strawberry or your cucumber, your tomato in the fridge from day 1 to 3rd day to 4th day to 5th day until it's spoiled. This hexanal, this specific chemical called hexanal, will increase through time, which makes it a good indicator or a biomarker for the spoilage prediction. Okay, so this is a really good question. So as part of Serenade project, we use these different parameters, the color, the texture, the physical, chemical, microbial properties, and we integrate them, all of them, to create these smart products, to create models using machine learning, and to create these eco-friendly, biodegradable materials or containers wherein you can put your fruits and vegetables inside the fridge, and it has this sensor that will detect when it's going to spoil. So from day 0 to the last day, and you will know the optimum time that you have to eat it before it becomes spoiled, which means spoiled, you cannot eat it anymore. So with the Serenade project, all of us, we are trying to develop, we're doing our best to develop this sensor, this packaging container, these biodegradable containers, in order to provide consumers and to help reduce food waste.
So when you ask about how this process, how I can improve, or this research could improve the food safety practices in the house or in the retail environment, when you look your food, basically you will not observe anything. So from your naked eyes, you cannot observe any microbial growth. Fungus can be observed when the food is tend to spoil. Mostly what are the spoilage you are observing is fungi, but not the bacteria. So for a consumer or maybe for the user, they are not able to differentiate. So in my research work, I am trying to look into a broader category from the fresh level of the food to until spoilage. So I will know how dynamically these volatile compounds are changing as a result of microbial growth. So I can able to differentiate the stages of food freshness, let's put it in that way. So it's easier for the, when it's coming to the application oriented, it's easier for you to differentiate the stages of food freshness. And when you think of the, when you think in the point of view like training or compliances, it's like for training persons or persons who work in retail environment, or maybe for the consumers, it's not really easy to describe about this microbiology process. They cannot understand it. It's quite you need some skills to understand the process. And so it's like a broad field, so we cannot explain. If I want to explain about the safety procedures, it's going to be longer, and for each microbial types it's different. So it's not easier to give a manual for a user to do such kind of things. And as I told in my previous answer, when you want to quantify or know the microbial spoilage, it's going to take time and need skilled workers. But if you are going to apply this kind of VOC detection method, it's going to be easier for you to detect, and for the users you can tell in a simpler way, like the signals coming from the sensors. So you can say like it's like in the road signals, what you're seeing. Okay, you're seeing green signals, it could be your food is fresh. If you're seeing yellowish, it's not, it's going to, in a transitioning stage, it's going to spoil it, and red is completely spoiled. They are going to easily understand, and I think it would improve your safety practices in a better way and way simpler.